to be bright awake now after lunch, because we have a real pleasure to welcome Marinka Zitnik. And I have known Marinka as our guest speaker today. I have known Marinka for a while now because we happen to come from the same country, but we live in very different parts of the world. Marinka is now an assistant professor at Harvard, and she is also a member of various boards and esteemed bodies, like the Board Institute of MIT and Harvard, and she's a faculty at Harvard Data Science. And Marinka, before she came to Harvard in 2019, was a postdoc at Stanford, and prior to that, she completed her PhD at the University of Ljubljana in Slovenia, hence our connection, although we don't know each other from those days. Marinka has been awarded and has achieved many accolades, and maybe I will mention just a few, like she won the Bayer Early Excellence in Science Award, and she's a rising star in electrical engineering and computer science at MIT and so on. So really, we are so honored to have you here, Marinka, today. Marinka's work is in machine learning, but in particular in application of machine learning for data in medicine and science in general. So I think that her talk today is going to touch upon that. And it is my great pleasure, Marinka, to welcome you here. Thank you.
Hi, everyone. Thank you so much, Mateja, for this kind introduction. I'm pleased and honored to be here with you today. And I thank the organizer for organizing this wonderful workshop. I just hope that I would be with you in person, but nevertheless, I hope that we'll have an interesting discussion today. So let me start by sharing my screen. Just a moment. Okay. Okay, great. So I hope you can see my screen now and you can hear me well. If not, please interrupt me. So today I will talk a bit about our research, and in particular, I will focus on ways and strategies to infuse structure and knowledge into biomedical AI. But before I go into detail and discuss this research topic, I would like to spend a couple of minutes discussing my career path and kind of some of the underlying motivation that guides my research. And so, as Mateja has mentioned, my research primary affiliation is at Harvard Biomedical Informatics. We are a very relatively new group that opened the doors or started the lab in December of 2019. Since then, we grew quite a bit. There are a number of PhD students and postdoctoral fellows that I'm very fortunate to be able to work with, as well as undergraduate and master students across different Harvard departments. And that primarily is the case because our research is, I would say, quite interdisciplinary. And so we have people on our team who have their core expertise and training in computer science, applied mathematics, but then also systems biology and bioinformatics, for example. We are located in the main quad of Harvard Medical School. There is where our primary lab is, and you can see this is a nice picture at the top that shows where our lab where our primary lab is. But then we also spend quite a bit of time on the other side of the river in Cambridge at the Broad Institute, and then in Harvard Data Science, where we are developing these new methods for a variety of biomedical data modalities. So in our research strategy is typically that we really probe thoughtfully and deeply on how machine learning in particular should be developed to address some of grand challenges in biology and medicine. I will later mention a couple of those. And so the way we are doing that is to identify what are problems in science and more concretely in biology that are on the cusp of being well positioned to benefit from machine learning. And so what I mean by that is that we are actively looking for opportunities where we see problems that are unsolved open problems in biology, but then there really are certain aspects that make them ripe to study from the lens of computational modeling and machine learning in particular. And so that often relates to the question of that we ask ourselves whether there are massive amounts of data available, that there are good strategies available to be able to evaluate the quality of the models that we'll be building. And so when we identify those problems in biology, we then ask questions about what kind of ML methods, need to be developed to advance that problem, and answering that question and developing the methods, then sometimes, but hopefully many times or often, allow us to advance the biological problem past state of the art and also create some new opportunities. And so very critically, we also conduct this in-depth work that then integrates our methodological advances with other disciplines. In our case, that means that we work often quite closely with biologists, with clinical researchers, sometimes virologists and other domain experts, to see how our methods can provide practical tools that can have tangible impact in real-world scientific discovery. And so what I just described to you, this kind of overall idea seems relatively simple and quite appealing. And so this might give an impression that the way research is done is what is shown in the left part of the slide. So this is what people think it looks like. So imagine we have this big idea. We just identified a super cool, exciting new problem that could be studied from the machine learning perspective that no one has studied before. So it's really potentially very impactful. And so the way people think of it and the way it typically looks like from the outside is that we have the idea, someone will give us the money or the funds or the resources, that can be university or foundations or National Science Foundation or NIH in the US, various funding agencies. Then that by getting resources, we now have the ability to build our model, to build our product based on the idea, to realize the idea that we had, and that will lead to overnight success, right? So that is how often then research projects are also presented. We have this linear pathway that starts from the idea and kind of ends at the overnight success. And so when I was a much more junior person than I am, I still consider myself a junior faculty, but when I was a PhD student or even a postdoctoral fellow, it kind of sometimes I felt frustrated because when you really work in the trenches of a research project, it does not look like a straight path, a straight line. So what it really looks like, in my experience, is more like what we see on the right part of the slide. So we have our big idea and we start a project. And then we work really, really, really hard. And we see these small promising results, but they're all preliminary results. We don't really necessarily are confident about them. So we are quite insecure, and we talk with various collaborators, and we go to conferences and present our preliminary results, and then solicit feedback from other experts in our field, meaning in our department, or perhaps at the conferences that we organize or we want to go to, and that can help us to further improve and refine the idea that we started with. And so that process can be lengthy and quite complicated, and there's lots of small successes and lots of small failures, and life happens in the midst of that. So what it seemed like an overnight success, then to everyone who is not involved, does not necessarily seem like an overnight success to those who are on a research team. So I think that this curvy line shown on the right part of the slide is much more realistic depiction of what it really looks like to work on a novel research project, where you are asking these novel questions that haven't been asked before. So the 2 points that I'd like to make here is that because of this nature of research where we have lots of small failures and lots of small successes before the problem is solved, it's really important to be able to persist and persevere. And no matter what, when there are challenges that arise, we need to learn from them and then try to kind of be better and persevere, and eventually things will happen. And so in my case, what has guided me and motivated me is intrinsically that I really like this kind of fundamental idea of science, which I think this picture on the left really summarizes it nicely. To me, it's just so fascinating that when we are doing science, the best part of science is knowing that for a moment there is you as a researcher know something that nobody else in the world knows, right? So you make a particular discovery, you derive a proof, or you train a model and make very promising predictions, or come out with a model of molecular system, or like a cell or a patient. And kind of that can give you some insights that really haven't necessarily been articulated ever before. And so that kind of, those moments to me are really the key parts that then I appreciate so much, that then help me go to discovery path from the start to what seems to everyone like a linear pathway or success of a project, but that's not necessarily how it really looks like. Okay, so with that in mind, I will kind of now spend a bit of time, or the rest part of the talk. I will kind of give you a few vignettes of a research project that we were doing in the lab that took that kind of trajectory or of a curvy, not straight line from the start and the end, but the success was there at the end waiting for us. Okay, so broadly speaking, my research group is really excited about modeling graphs or networks and thinking of them as a universal language for describing complex systems. And biology is really an example of very complex system where we see graphs and networks at many different scales, from the scales of a cell, where there are many different molecules like proteins within a cell that interact with each other, that interact with other biological elements, then within an individual that has some diseases that might again interact and could depend on each other, and takes medicines that can influence the behavior of those proteins and genes that were altered by the disease. And finally, networks really exist also at the level of communities or entire healthcare systems. So we see these multi-scale network representations and complex networks that really pervade much of biology and even healthcare, I would say. And so we are really excited about those problems, and we're developing then models that can learn and reason over those network data set. And so the kind of models that we're developing nowadays are called graph representation learning models. So this is an emerging, very quickly growing field that is a leading paradigm for training powerful deep learning models on networked interconnected graph structure data sets. And we have seen that graph representation learning has led to many areas of success in biology and beyond. So for example, it led to these insights in network biology that I will mention today, discovery of new drugs that are safe and effective. But outside of biology, graph representation learning has really been a powerful paradigm in recent years for developing recommendation systems, for simulations in physics and modeling of evolving graphs in physics, or reasoning about scientific knowledge and identifying what are gaps in knowledge that science or society more broadly could fill. And so graph representation learning as a field has been powered by a particular class of algorithms or methods that are known as graph neural networks, that I think some of the other speakers have already mentioned. So very briefly, I'll kind of outline the key idea here on this slide. So graph neural networks are neural architectures that are very flexible and can work with any relational graph structured data sets. So you can think of them as a class of models that is complementary to other prominent classes in deep learning, and those are primarily neural networks for analysis of images, which one can think of some form of grid structured regular graphs, and recurrent neural networks and transformers for modeling of sequential data or language. And so graph neural networks, complementary to those two very prominent classes of methods, allow us to train deep learning models on a graph structured data set. So the way this is done is we start as the input on the left with the graph. So here's a small graph where nodes are actually different genes, and we have edges connecting those nodes. And so graph neural network is then a nonlinear function f here that is specified by its architecture. And what it does, it will take each and every node from the input graph, it will map it to some d-dimensional vector embedding or vector representation, which we typically term embedding. And so the idea is then to optimize parameters of that function f such that when 2 points, 2 nodes from the input space are considered to be similar, to have similar network neighborhoods, they will be positioned very close together in the embedding space. And so the idea is then to optimize the embedding space, infer this compact embedding space such that performing linear algebra operations with those learned vectors will reflect topology of the input graph. And so graph neural networks are very flexible because they allow us to specify a variety of different functions f through a mechanism that we call neural message passing that can then operationalize different meanings of when 2 nodes have similar local neighborhoods. And so this gives rise to a large number of different kinds of methods and architectures that all operate on that idea of examining local neighborhoods of nodes from different perspectives and embedding them into some low-dimensional compact space. So GNNs have been shown to be successful and very useful across a variety of domains. And in my group in particular, we are looking at across biological, medical, and knowledge networks. And we have spent quite a bit of time to try to demonstrate the utility of GNNs to look at patient networks. So those are networks where nodes are patient and edges indicate how patients interact with each other, or they indicate friendship or familiar relationships. And then networks at the molecular level that describe how biological processes depend on each other within a cell, or what kind of networks are altered in a disease, or gene interaction networks, or more recently also single cell networks. So we really found that us and others, many others, that graph neural networks trained across biological, medical, and knowledge networks are very useful class of algorithms that can lead to exciting new insights. So with that in mind, my plan for today is to talk about 2 vignettes and 2 topics in particular. So the first part will talk about ways to infuse structure into biomedical AI, or more concretely, I will, from the algorithmic standpoint, I will talk about few-shot learning for graphs, and from the application standpoint, we'll see how few-shot learning for graphs and methods that we have developed have allowed us to identify promising therapeutic opportunities for emerging pathogens. In the second part, I will focus more on modeling networks from the mesoscale, from the standpoint of subgraphs, rather than looking at individual nodes and edges. And I will present a recent algorithm that we developed called subgraph neural networks. And if there will be time, also talk a bit how that algorithm has helped us to develop AI assistant tools for diagnosing disease. Okay, so let's begin with the first topic. So the motivation for this research, or this line of research, has been the following observation, or the question that we asked ourselves. We were wondering, like, we really want to ask fundamentally what makes biomedical data so unique? Why do we need to develop specialized ML models for biomedical data? Or in particular, what are the kind of challenges that are unique to biomedical data that we need to address and really require novel methods? And so one observation was that for many problems in biology, there is very little label data available. And so that's a challenge because recent breakthroughs in AI have primarily depended on lots of labeled data that were then used as inputs to train supervised learning models. And so it has been really shown that large diverse data sets can lead to broad generalization. That has been shown very successfully by many in computer vision, for example, or in natural language processing. But what I would argue that for problems in biology and biomedical discovery, they're very challenging because often when we really ask a new question, it requires us to have a model that can generalize to a new phenomena, that can generalize to data that was not seen during training of the model. And so that is a challenge because prevailing methods for graphs require abundant label information, but labeled examples are typically very scarce for problems that are at the frontier of biology. And if you remember at the beginning of our talk, I said that we often look at problems at the cusp of biology that are really mature enough to be able to benefit from ML. And so examples of that kind of problems that are very exciting, but for whom labeled examples can be scarce, include working in drug discovery, identifying modeling novel drugs in development, or modeling emerging pathogens, or trying to identify treatment opportunities for hard to diagnose patients or patients with rare diseases. So visually, this problem can be depicted as follows. What prevailing methods assume is that the data, how it looks like this graph on the left part of the slide. We have a graph. We want to predict some properties or aspects of nodes, and much of that graph is already labeled. So for most of the nodes, we already know what they do. And so the goal is to leverage that information to label and predict the labels for the remaining nodes. But what really happens in real world is it does not look like what is shown on the left part of the slide. It's shown what it's look on the right part of the slide. We have these vastly unlabeled large graphs that is unlabeled. And there are just a handful of nodes that we really know what they do. We just have a handful of nodes that are colored representing labeled examples, and much of the rest is just black or dark or gray matter in this example, right? So, but that gray part is exactly what science is interested in, because we want to advance science, rather not just recover or identify again what is already known. And so that motivates us to be thinking a bit more closely about opportunities and ways to do few-shot learning on graphs, which is a machine learning term that describes the problems of learning with very few limited labeled examples. So here's one slide of background on few-shot learning for those who are not familiar with it. And I will show an example on images rather than graphs, just to illustrate the point. So few-shot learning is a particular concept that belongs to an area that is known as meta-learning, and in meta-learning is a part of supervised learning, but it refers to the situation where we have only a few shots, a few labeled examples available for learning. And so typically we would say that we have k-shot learning if we have k shots, k labeled examples per class. And so the way few-shot learning is done is that we train our models in a slightly different way than what standard supervised learning would do. So we do the following. We have two stages of training our models. The first stage is called the meta-training phase. The second stage is called the meta-testing phase. In the meta-training phase, what we will do is we will really try to teach the model to be able to very quickly train classifiers while having only small number of labeled examples available. So to do that, we will prime the model through a number of small training tasks that we'll define. Each training task has only a handful of labeled examples per class. So in this example, training set 1 has 2 labeled examples, and the task is, can we train a classifier with just 2 labeled examples available, and there are 3 classes that we want to categorize those images into. And so the goal here is that we will train the model such that we will be able to quickly generalize from one training task to the other, each of those training tasks were designed to be quite challenging and very difficult. And so the reason why we are doing that in the hope that once we go into the testing phase, when we will really be solving the new tasks that we're interested in, for which very few labeled examples are available, the model will also be able to very quickly generalize and make and produce a good, accurate classifier with just a few labeled examples. So in this particular case here, a test time, we would be asked to build a duck versus dolphin versus chicken classifier. And with few-shot learning, that would be possible to do having only 2 labeled examples for each of those 3 classes. And so what our research the research that we did in this area was that we thought very carefully about how can we design few-shot learning strategies and methods that would operate not on images, but would really be able to learn with few labeled examples with limited labeled information on graphs. And so the algorithm we developed that I'm highlighting today is called GMeta. It is a few-shot learning strategy for graphs that represents a general framework and that is also theoretically grounded, as I will show later, and we published it at NeurIPS last year. And so the idea here is that we adopt a similar strategy of training as I showed on the previous slide. So in the meta training phase, we'll define a number of training tasks. Each training task will consist of a small number of labeled examples and and we will train a graph classifier to leverage those labeled examples to accurately predict labels. And so in the meta testing phase, when we will be tasked with classifying nodes into a new set of labels, Y star, the model will be able to very quickly generalize to those blue labels, even though only 2 labeled examples are given in this case. And so that's what GMeta will allow us to do. To formulate this problem a bit more concretely, I will just focus on one particular instance of few-shot learning that GMeta can handle, and in the paper we describe other settings as well that GMeta can be used for, so that it really represents a general framework that can be used in any graph representation learning environments. And so in the simple case, the problem here is that we have a graph G, we have some distribution of labels that we denote Y. And so the goal then is to train a classifier, or which we'll call meta learner, that can very quickly adapt to an unseen label set, Y star, by learning from tasks that have other label sets, Y, but there is no overlap between those 2 label sets. So visually, what that means is that we have graph G where there might be an abundance of yellow labels, but there are very few blue labels. And we really are interested in being able to accurately predict and identify nodes that have blue labels, but we cannot do that because there's so few blue labels. So what we will do is, during meta training phase, we will really train the model on a variety of training tasks defined on yellow labels in such a way that the gradient of the model will very quickly generalize then to blue label set. So the key idea to do that is that what we are doing is to thoughtfully probe local neighborhoods of those nodes in the Y label set and derive something that we call subgraph signature function, which is essentially a differentiable function that can allow us to describe every training task. And so what we can do then is to take those sets of nodes that describe a particular training task, design a graph neural network app that can map those nodes based on the structure of the local neighborhoods to d-dimensional embedding space, and then derive a subgraph signature function that describes a particular task. That subgraph signature function is a mechanism, it's a kind of information that is transferred from one task to the other, to the third one, to the fourth one, and so on. So essentially means the more tasks the model is asked to solve during the meta training phase, the better subgraph signature functions it will learn. And so that the final subgraph signature function, it will be used effectively to initialize the model in the meta test phase to very quickly then just fine tune the model on a limited number of labeled examples and get a good classifier. So why are we looking at local subgraphs or local structure around those small number of labeled nodes from label set Y in order to generalize to Y star is a very good question. So and that's a question that we also ask ourselves, and we answer it by studying the influence of one node in the graph on the target node that we want to predict label for as a function of network distance between the two nodes. And in the context of graph neural network architecture, what we'll be able to show is that the influence of a node on the target node can decrease exponentially as we go further away from the target. And so that is true for prevailing graph neural network architectures today. So long story short, the implication of that is that if we look and only examine local subgraphs around target nodes, we will not lose much information. It will those local subgraphs will contain all the relevant information and preserve near the same feature information as entire graphs. That's really important because it makes our algorithms scale to very large data sets. Conceptually, you can think of the value of subgraphs in the following manner. So when we train machine learning models on graph structure data, we typically leverage two kinds of sources of information. The first source of information and the first kind of strategy is that of label propagation. So here we are assuming that nodes with the same label tend to be close to each other nearby in the graph, and therefore we can propagate those label information in order to classify, in order to make predictions. The other complementary view that is exploited by machine learning models for graphs relates to structural similarity. Here, the idea is that nodes that have the same label will not necessarily be close to each other in the graph, but they might have similar network shapes in their local neighborhoods. So imagine a graph that would consist of nodes that represents people in a particular organization, and edges indicate who sends email to whom. In such a graph, you might find secretaries who would be nodes that have the same role or the same label, be actually having the same label, but very far away from each other in the graph, if those would be secretaries, for example, from different departments in organization. So the reason why we're using local subgraphs in GMeta is then the following. When label information are scarce, propagating labels will not be sufficient. There are just too few labels one would have, so we cannot effectively rely on using only examples of other labeled nodes to train a classifier. It's just not enough data where we just have 5 or a handful of labeled examples. And so we need, because of that, we need to much better leverage structural similarity when we have limited labeled examples available. And so that's exactly what local subgraphs do, because they characterize the structure, motifs, and network shapes around target nodes, and they define those subgraph signature functions that are signatures of that structural similarity information. Okay, so with that in mind, I will now change gear and show you and describe to you how GMeta algorithm and few shot learning graphs have been really powerful and useful to us in one particular project related to drug development. So the project that I'll be highlighting describes the work that we have done within the COVID-19 task force, and primary part of the work that I will highlight today was done between March 23rd last year and June 15th last year. So that's just a span of 3 months. And during those 3 months, we were able, with a team of our experimental collaborators, biologists and virologists, we were able to compress years of or months of work to weeks or even days in some cases, in an effort to identify promising therapeutic opportunities for an emerging pathogen. And so I really like this example, not only from the perspective of how new algorithmic development can lead to tangible practical results, but also from the sense of really demonstrating concretely how machine learning models can practically help accelerate and expedite the development of novel medicines. That is particularly important in contexts such as pandemics that demand safe and effective therapies developed at unprecedented speed very, very quickly. And it is in sharp conflict with traditional process for developing drug discovery that is very resource intensive. It's not only expensive, but it takes on average between 11 and 16 years to develop a new drug from scratch. And so that traditional iterative development, experimental clinical testing of new drugs is really not feasible when we're faced with a pressing healthcare emergency. So in this project, we really want to be very kind of practical in the sense of how, in the sense of answering question of how can we compress now years of work or into months or weeks through the use of AI automation and novel data resources. Okay, so let me tell you what we did. So fundamentally, the task or the problem, the question that we want to answer, if we want to inform drug discovery and development, and in our case, repurposing of existing drugs for this emerging pathogen, is to have a model, a predictor that will be able to predict what drug would treat what disease. So the goal is to have a classifier that will operate on this large multi-scale biological system that I've been describing at the beginning of the talk, and will be able to predict for a given chemical compound or for a given drug that we already have on the market but was not designed for a new pathogen, predict if what kind of disease that compound might treat, or that would be useful for, in terms of it would either treat symptoms or the root cause of the disease, or it would alleviate some of the issues that the disease causes. And so in our case, the disease that we were interested in was COVID-19. And so working on this problem back in March 2020, it was very different from working on this problem today. So in March 2020, very little information about COVID-19 was available. And so the first question that we had to ask is, how can we even computationally represent that disease in an order that would be amenable to machine learning analysis? So in our case, we relied on the data generated by Gordon et al. From UCSF. And what they have done is they looked at the virus, and they have expressed 26 out of 29 SARS-CoV-2 proteins, and then use a mass spectrometry, a type of biological experiment, to identify what are proteins in human cell that virus attacks or binds to. And so that set of 332 human proteins was to us what we did, computable unit, which we refer to as viral disease module or COVID-19 disease module. And so then the idea was, if we overlay those set of 332 proteins onto the network that describes how human proteins interact with each other in the cell, that can inform us and identify, well, what are those drugs that will likely mitigate the effect of the virus, in the sense that the drugs would fix and alter the proteins that the virus has attacked. And so that means that we need to have a model that will reason about the relationship between this viral disease module shown in gray and a large number of drug representations or drug modules that are defined as these modules that in the human protein interaction network, where each module is defined on a set of nodes that a particular drug will target, a set of proteins that drug will target and change their behavior in an effort to alter the disease. So the data set and experimental setup here was then the following. We constructed this based on the idea I showed on the previous slide. We constructed a COVID-19 repurposing knowledge graph, which had the underlying human protein interaction graph. That is what we started with. So nodes are proteins, edges are physical contacts between proteins. On top of that, we overlay a large number of drug modules, all drugs that were approved in the US at the time, and every module is defined as is a part of the PPI network induced on a set of proteins that the drug targets or binds to in human cell. And then we had the disease modules. We have COVID-19 disease module, as well as we constructed the disease modules for many, many common diseases that already have some treatments and are much better understood from the biological medical standpoint. Given the data, the goal was that the following: given common diseases and treatments for them, can we learn from those other diseases and identify what are disease treatment mechanisms in an effort to identify candidate treatments for COVID-19? And so here's where few-shot learning comes into play, right? COVID-19 at that time was never before seen disease. Of course, we didn't have any example of successful treatment for COVID-19, meaning no labeled examples were given. And so that's the hardest setting in few-shot learning, that is known zero-shot learning, where essentially you need to train a classifier, but you don't have any labeled example for that classifier. So what can you do? And so zero-shot learning and GMeta helped us to leverage information about common diseases and treatments for them and transfer that information very effectively to this new pathogen that was emerging pathogen never seen before. So the way we practically operationalized GMeta was the following. In the meta training phase, we defined a number of training tasks. Every training task was designed for a different disease that already has some known and successful treatments. And in that stage, the algorithm was asked to quickly train classifiers that would identify what drugs that will correctly predict what drugs are effective for treating the disease. Disease 1, disease 2, and many others. And so in the meta testing phase, we took that GMeta model and be able to directly use it for COVID-19, even though for COVID-19 there were no examples for successful treatments. And so that's what GMeta allowed us to do. Training training those GMeta models, the first question we asked when we trained those models was, how good are those models? And so we, the first idea we had to answer that question back in March 2020, or that was in April 2020, was to look at drugs that were studied in clinical trials at that time. Those drugs are not those drugs that would necessarily work successfully, but the medical community thought of them as promising or worthwhile exploring. And so what the results of that experiment are shown on the left part of the slide, where we are showing true positive rate as a function of false positive rate. So the better the model, the closer the line, the accuracy line would be to the top left corner. And so what we can see here that GMeta was able to outperform many other techniques, ideas that we try out, including those that are based more on classical machine learning models and network science principles, such as diffusion models or network proximity algorithms and manual feature engineering. And so that has motivated us to really use this model for discovery and informing biological experiments. And so as part of the COVID-19 task force, our collaborators, virologists, took predictions, top-ranked drugs that were produced by our ML models, and they have validated and screened them in wet lab. And so that was done in National Emerging Infectious Disease Laboratories in the US. That's a level 4 laboratory, which means that it's a laboratory that can work with live virus and cells affected directly by the virus. And so the results of those experiments, without going into details, were that 918 compounds that were predicted by the model were screened for efficacy against SARS-CoV-2 virus. This was first done in African monkey cells and then later human cells. And the result of that was that by screening drugs, top-ranked drugs in human cells, we obtain a near 62% success rate, meaning that 62% of drugs that were tested, that were predicted by the model and were tested, turned out to really inhibit viral growth. In contrast, if we would not be using any ML modeling and those experiments would not be guided by AI models, we would only obtain a 0.8% hit rate, which means an order of magnitude worse performance without using ML models. And so that was really, really exciting to us. And it certainly demonstrates how ML models can really help us expedite this important problem of discovering safe and effective medicines. But on top of that, we also wanted to know what is, if anything, is common to those drugs that were predicted by the model to be effective or to induce, to inhibit viral growth, and that were also validated by experimental screen. And so what we found is that 76 out of 77 drugs that were predicted and shown to successfully reduce viral infection were the kind of drugs that don't work by directly affecting or targeting those proteins that the virus attacks. Instead, those drugs bind to some other proteins in the human interaction network, and then those effects propagate along edges of the network to eventually reach the proteins that the virus attacks. So that means that those drugs rely on network-based actions, and importantly, they could not be identified by other strategies for drug repurposing, such as molecular docking. And so to me, as someone who works on the methodological or theoretical side, what was really exciting is to see how as a computer scientist and method developer, we really can have impact and influence on a completely different discipline, and in particular how networks are really a universal or language for modeling these complex systems, and modeling that effectively can yield certain insights that we could not obtain otherwise. We could not obtain and identify without really taking a network-based approach to research here. Okay, so that concludes the first part of my talk, but since I just noticed that I'm running rather late, I will perhaps spend just a couple of minutes on the second part of the talk to give to kind of give a quick vignette of that part, and then conclude to leave a few minutes for questions. And so this, in the second part, we will change gears a bit. So we will go away from molecular biology and tackle a different problem that is closer to patients and medicine. And so in that context, we will kind of change our problem formulation a bit. And in this context, our problem formulation is that we will work and want to design models that can learn on large networks or graphs, where those graphs are not graphs of proteins, but are graphs of that describe different kinds of entities and medical concepts and represent patients. And so what I'm showing here on the left is a small graph that has a number of subgraphs overlaid on top of it. We have one red and two yellow and one green subgraph. And essentially you can think of each of those subgraphs as a patient. So we would have 5 patients, and every patient would be described by a set of disease diagnostic codes and drugs that the patient is taking, and what kind of medical procedures the patient had. And so disease diagnosis problem can then be formulated as a question about taking those subgraphs and be able to predict labels for those subgraphs, and those could correspond to patient outcomes. Now, to be able to do that, what we need to do is we need to model effectively those subgraphs. So to do that, we can formulate the problem as that once we have those graphs, so the goal is to learn now not individual node embeddings, but subgraph embeddings, such that the likelihood of preserving subgraph topology in the learned embedding space will be maximized. In other words, if we have two subgraphs on the left, on the input side, that have similar subgraph topology, then we want to learn such subgraph embeddings for them that they could be embedded close together in the embedding space. And so the method that we developed that allows us to do that is called subgraph neural networks, or sub-GNN. We published it at the last NeurIPS. I will not be able to go into detail, but I will just kind of conclude with a practical example of how sub-GNN has then been able to help us diagnosing diseases in patients. And so this question, as I said, is the question of asking how can we leverage the set of diseases, phenotypes that the patient has in their medical history in order to model diseases better and identify what is the likely disease that a patient might have. In particular, for those diseases that are otherwise very hard, or for those patients that are otherwise very hard to diagnose because the symptoms that they have and their medical history are very different from what we would expect based on medical knowledge. And so this can be motivated as a problem that we have a graph with those symptom subgraphs that represent different patients. And given that information, then a subgraph neural network can allow us to prioritize what are the most likely diseases, that a particular patient might have, which in machine learning language would be formulated as predicting labels for subgraphs and producing a ranked list of those labels. And so using subgraph neural network model for this kind of problem is again very helpful. We can demonstrate that it outperforms baselines in this task of subgraph labeling by an average of 77% on synthetic data sets, and achieved even stronger performance on real-world data sets. That it has motivated much of our ongoing research nowadays when we are interested in modeling patients that are hard to diagnose and assisting in diagnosis of those patients. So with that, I will wrap up for today. All the papers, code, and data set that I have mentioned are available at our lab website. If there is time, I'd be happy to take a question or two. And of course, also feel free to reach out to me via email. I'll be happy to follow up on that. Thank you.
Thank you very much. Thank you, Marinka, for that really interesting talk. So I will first look around the room if there are any questions in the room, and then I'll look online and see if there are any questions in the chat. And while people are getting courageous to voice their plentiful questions, I will ask Marinka, have you thought, when you construct a graph neural net, you were using PPI networks, I presume, to inform also what the weights for the structure of the graph. Have you thought about using domain knowledge, like, for example, using gene ontologies or something like that, if you want to use genomic information in order to initialize or inform the weights for your edges on your graph?
Yeah, so that's a great question. Absolutely, we are integrating these additional sources of biological knowledge to inform graph neural network architectures. And in particular, that is important from several different perspectives. One is that by using domain knowledge to induce the right relational biases, we're hoping to be able to obtain more accurate models. The other important motivation for this part is that in science and in biology in particular, and we are not only interested in accurate models, but sometimes we are even willing to sacrifice accuracy for models that are more trustworthy or more interpretable, or can provide not only point-wise estimates for a predicted label, but can also identify part of the data or that drives a particular prediction, that can provide us more actionable mechanisms that can inform downstream experiments. So that is very, very important for if you really want to use those models for any kind of new scientific discovery. And so that's something that we are actively working on. I didn't talk about it today in this talk, but questions related to obtaining more actionable predictions that can lend themselves directly to downstream experiments by integrating additional knowledge that can lead to more interpretable models is something that we are very excited about.
I'm very happy to hear that. Any more questions or thoughts, online or in person? Yes, we have a question.
I was intrigued when you were trying to learn possible drug candidates, this one for COVID, that not only did you have very little data, but the data you had, you didn't really know how good it was. I think what you said at the end, where actually the possible drug candidates that you found, it was all network effects and it wasn't the sort of landing site effects, which might be the more obvious route. So that the data that you're sort of training on, sort of, there's lots of human error involved, there's lots of, there might be other candidates out there that are better than the ones that are in your training sets and things. I wonder if you could comment a bit more on that, how you dealt with the sort of varying quality of data.
Yeah, so that's a great question. In general, biological networks are incomplete and messy, meaning that many edges are missing because there simply were not experimentally probed yet. And the kind of data that is there might be erroneous and represent false positives. So in addition to that, in the context of COVID-19 applications and research, a major factor is also investigative biases, where we see that certain nodes in the network or sets of nodes are just much more often investigated because of interest in biomedical community or the ease with which experimental work can be done, and then this artificially creates more edges around certain nodes than we would expect there to be, for example. And so the way this was tackled, it was by very careful pre-processing, and then in particular in our models we had controlled. We had negative controls, and those negative controls in our case were designed based on common diseases that already have some approved treatments for. That's what we have used as a way to identify what biases in the networks we have and to mitigate those biases. Regarding those discoveries about networks drugs that you asked, so those drugs that were identified, that new class of drugs that we refer to as network drugs, are drugs that were indeed effective and were inhibited and did inhibit and reduce viral growth. And so the interesting part of that is that it's not that networks did not work well. In contrast, it was that networks were able to identify this set of drugs that were surprising, yet they were shown to be effective in the sense of inhibiting or attacking the virus, and they have these network mechanisms that are surprising relative to what biology would know, and so that might be, and sort of those indirect effects that we think of them as being indirect effects might actually be now direct effects if the underlying data was complete, but it was not because of natural biological variation. the fact that the models can pick those drugs up is really exciting. It shows the power, it indicates the power of ML models for graphs here.
Thank you very much, Marinka. I'm afraid that's all the time that we have now. Let's thank our speaker. It was really wonderful to hear about your cutting edge work. Thank you very much.
Thank you so much for inviting me. It's been great to be here.
Now, I'm hoping that we have Caroline.
I'm here.
I can see you, Caroline. Caroline or Carolina?
Both is good. Yep.
Okay. Would you like to test out the sharing of the screen?
Let me share my screen.